Hi guys, good morning. This is Dr. Renz Edwinderla again. And our topic for today is all about the embryology of the limbs and the muscles. Now, in our last lecture, we talked about the processes of the developing embryo, and the last process is the formation of or the stage of gastrulation. This is where there's formation of the trilaminar embryo. Trilaminar meaning there's the three germ cell layers formed. So we have the ectoderm, the mesoderm, and the endoderm. Now, um, let's focus ourselves on the mesoderm for that matter. So this picture is a cross section that shows the three layers. So for example, this is the amniotic cavity. So this is the yolk sac. The blue one is the endoderm. The yellow one is the blue one is the ectoderm, the yellow one is the endoderm, and the red one is the mesoderm. So for the mesoderm, it is also divided into parts. Now for our topic, the development of limbs and muscles, let us focus ourselves on the mesoderm. So the mesoderm has three parts: the paraxial mesoderm, the intermediate mesoderm, and the lateral plate mesoderm. So here's a diagram, somewhat a preview of the things that we're going to discuss. Now for the one closest to the neural tube, as you can see here in the notochord, is the paraxial mesoderm. So this has two sides, bilateral. So the paraxial mesoderms will give rise later on to the somite, which will form the head, and it also form the sclerotum, which will make the vertebra and ribs, and the somite will also become the myotome and the dermatome, the skeletal muscle, and the dermatome, the dermis, and some of the skeletal muscles. Now for the lateral plate mesoderm, this is divided into three: the splanchnic, somatic, and extraembryonic. But focus yourself on the splanchnic and the somatic. The intermediate mesoderm will not be discussed thoroughly here, since they will only form the development of the kidneys and the gonads. So for the development of the muscles and the bones, the intermediate mesoderm has no direct contribution. Before we proceed any further, let us get a clear grasp on what happens on the fetus. So there's an event called embryologic folding that happens around the third week to the end of the fourth week. And this involves two types of folding: the horizontal fold and the median fold. Now I will share to you a video because it's easier to understand if you can see through animation. So here's the video. So let's review what we have so far. So remember, I told you to focus on the lateral plate mesoderm. So we have the somatic and the splanchnic mesoderm. So during folding, they will also go with the structure that they are mostly related with. So the splanchnic mesoderm went with the endoderm, and true enough, they form the gut tube. And you can see here, the somatic mesoderm, it went with the ectoderm, and they form the lines of the abdomen. So in later development, the somatic mesoderm will form the muscles, and the somatic mesoderm that went with the ectoderm will also form the dermis of the skin. That's what happened between the two branches of the lateral plate mesoderm. Now let us focus ourselves on the paraxial mesoderm. Remember that the paraxial mesoderm will give way to the formation of the somites. Now, somites are segmented uh, paraxial mesoderm. So you can see here in this picture. And each somite is divided into two parts. The sclerotome, which is the ventral part, and the dermomyotome, which is the dorsal part. The sclerotome, which is in closer relationship to the neural tube and neural crest, will help make form the vertebral disc or the spinal column. Now the next task of the embryo is to decide where to put the limb buds. And it's not just any decision that he can make, it must be regulated. Now, with the help of these genes, the limb buds will form at a specific area of the embryo. So these genes that decide the position of the limb buds is the Hox gene. And the genes that will decide which is the upper limb buds and the lower limb buds will be the TBX genes. After the embryo has determined where to put the limb buds, there's going to be a thickening of ectoderm in that area. Now, with the thickening of the endoderm, this will form this one, the AER, or the apical ectodermal ridge. And through this elongation, another structure closely related to the ectoderm will follow. So remember that what we discussed before, the structure that is closely related to the ectoderm is what part of the mesoderm? The lateral plate mesoderm, specifically the somatic plate mesoderm. The splanchnic mesoderm join the endoderm forming the gut tube, while the somatic mesoderm join the ectoderm to form the dermis and some other muscles. Now in this case, the somatic mesoderm will also join the ectoderm in forming the limbs. So through the AER will create a somewhat proliferation. The mesoderm will also crawl inside or will also get inside. And this will help formation of further structures. Now let us focus on the AER. In this diagram, we can see that it is closely related to the mesoderm of the newly formed bud. The AER will produce growth factors, specifically the fibroblast growth factors that will influence the mesoderm to proliferate. Now the mesoderm will proliferate and will help continue to make the limb grow or make the limb extend. Now let us take a look at the AER more closely and how the limbs develop. The area closest to the AER is greatly influenced by the growth factors. So the mesoderm in this area is greatly influenced by the growth factors. So it's undifferentiated. So it's undifferentiated. Its only function is to proliferate. So this area, this one in green, is the undifferentiated zone, and its only task is to proliferate. So it makes the limb grow longer. As it makes the limb grow longer, the one farthest from the undifferentiated zone will become the differentiated area, or it will now differentiate. Since it's far from the influence of the growth factors, it will now start to differentiate. So at first, as you can see, it's from proximal to distal. So this one colored in blue, it will elongate, and then the formation of another area, here one in pink, then again it will proliferate, and then the last is the formation of this yellow area. So this yellow area is what we call the autopod, the pink area is the zygopod, and the blue area is the stylopod. So these are just embryological terms or names for the parts of the upper extremity. So for the autopod, this is the hands, the zygopod, this is the forearm, and the stylopod, that is the upper arm. So as it elongates, the nearer it is to the AER and the influence of the growth factors, it is not differentiated. But when the limb grows and it's the cells or the tissues that are created and it's now far from the influence of the AER, it will now differentiate. Now, for the blue one, the humerus will form, the radius and ulna will form from the pink one, and later on, the fingers will be formed. I want you to look at this picture. At some point, some areas of the apical ectodermal ridge will die or apoptosis will happen. This is due to the retinoic acid and the bone morphogenic protein or the BMP. So specific areas of the AER will die, and this is, you can see it here, but some areas is still progressing or it's still maintaining. So as you can see, fingers are now starting to form. So the areas where the AER is maintained, that is now making the fingers. Now the area where the AER regresses, it's the space between your fingers. It stops. Now the AER, all of the AER will stop influencing the proliferating zone. It will stop producing the growth factors, and eventually this part of the hand, instead of proliferating, because the drops in the levels of the growth factors will go down and there's no influence of the growth factors anymore, what will happen is that these cells will stop proliferating and will now undergo differentiating. So what will they differentiate into? First, they will differentiate into cartilage, and then this cartilage will become bone. Same with the humerus. At first, it will become cartilage, then later on, it will become bone. It is what you call endochondral ossification, the formation of bones from cartilage. Now that the limb is formed from the AER, the fingers are formed, the bones have been formed from the lateral plate mesoderm, I want you to look back at the paraxial mesoderm, where, remember the somite? The somite is divided into two. The ventromedial part will become the sclerotome. The sclerotome will become the vertebra and the ribs. And later on, look at the other part. It will become the dermomyotome. Now the dermomyotome will have the myotome and the dermatome. Now, remember that the limbs, the bones are there from the lateral plate mesoderm, but muscles is still not there. Now, the myotome from the somites will migrate into the limbs, into the from distal to proximal. It will migrate. And also, aside from the limbs, the synditome from the somite, particularly the sclerotome, the synditome will also migrate into the limbs. The synditome will become tendon, and the myotome will become skeletal muscles. So now we have the complete picture. We have a bone, we have tendons, and we have the muscles.